Okay, so this is the second half of chapter 17, which is about the viruses. The first half is already online. It discusses the lysogenic lytic cycle, the general structure of viruses, a little discussion about viruses versus living cells, and for you to think about those characteristics of life and what viruses have, because they do have a lot of those characteristics, and then what they don't have and why we don't consider them living generally. Okay, so we talked about phages before. Those are viruses that infect bacteria. So the ones that inject their DNA that look like little, sort of screws or alien-like things here with the head, those are all bacterial infecting viruses. And so now we want to talk a little bit about animal infections and plant infections, and it's going to be very cursory. You can take a virus epidemics class, epidemiology class, very interesting later in your scientific career if you so choose, and you'll learn a lot more about this. So really, with animal viruses, we classify them by two criteria. The first one is how the viral genome, what it's made of. So remember we talked they can be made of single or double-stranded RNA or DNA. We see a lot of animal viruses being RNA versus RNA viruses for other organisms. And then the presence or absence of an envelope. And remember, we see the envelope is really important. Way back here for entering the host cells, right? It has all those glycoproteins of both host origin and viral origin on the outside, and then it has its capsids with its nucleic acids, this genetic material on the inside of that. So a lot of animal viruses have envelopes, and so here's just an example of different types of animal viruses you've probably heard of. So we classified them by double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA, and then some that are using RNA, single-stranded RNA for synthesis and for RNA synthesis, and single-stranded RNA for DNA synthesis, and this one's the mRNA. So all these are RNA, single-stranded, but they use the RNA in different ways to replicate inside a host cell. And then you can see here envelopes versus not envelopes. So, I mean, you've probably heard of a lot of these adenovirus, right? That's going to be your respiratory infections, non-bacterial herpes, other kinds of herpes viruses, the chickenpox, the varicella zoster, which also causes shingles, Epstein-Barr, which is mono or mononucleosis, your smallpox, which fortunately we don't have to deal with anymore, and those are all double-stranded DNAs. The herpes virus has the envelope. You've probably also heard of maybe coronavirus. That's the one that causes the SARS. And rhinovirus is a picornavirus, which is the common cold one. Also hepatitis A, you get vaccines against A. C is also in the group as West Nile. We have to worry about that here in Houston. And then Ebola, you might have heard of that, pretty severe virus. They've made some movies about Ebola virus. And then, your flu, measles and mumps. And here would be the HIV virus, as well as some viruses that may cause leukemia. Some viruses actually can cause cancer depending on where they disrupt the genome, but not all. But you've heard of HPV, right? Human papillomavirus. So that's cervical cancer. That's the one they have a vaccine for now. And then also sometimes Epstein-Barr rarely causes Burkitt's lymphoma, which is also a type of cancer. And we'll talk about that a little bit when we talk about cancer and cell division chapter. So these viral envelopes, useful for entering the cell if they're present. They come from plasma membrane of the host cell when the virus, the capsid, and the nucleic acid comes off of the host, buds out of it. You see the host cell is going to have some of its glycoproteins on the outside, and it'll have some glycoproteins you can see being made here that are actually encoded by the virus itself. Sometimes actually the envelopes can even be derived from the nucleus of a cell. And then sometimes they'll have a double membrane. It just depends on where the virus, or at least helps the virus infect different sections of a cell. So here's the general schematic for how an RNA virus would infect an animal cell. So RNA, single-stranded RNA is the genetic material. This one has an envelope, which most RNA viruses do. The envelope fuses with the cell membrane. The virus ends up being inside with its capsid and its nucleic material. So here, in this case, we have the viral genome, the RNA, being used as a template to make more RNA. Hold on, let me go plug in my computer so I don't lose you guys. Okay, I'm plugged in now. Okay, so in this case, the mRNA is used for two purposes. Both are going to be coding for proteins. And remember, the proteins are made using all the the host materials. So they're ribosomes, they're nucleotides, they're amino acids, all that stuff. And so some of the mRNA is going to actually be read by ribosomes, and it's going to dump that protein into the ER, because it's going to end up being these glycoproteins in the cell membrane. So that has to go through the secretory pathway, the endomembrane pathway. They don't get secreted, but they get put in the membrane. And some of the other mRNA is read by different genes. Make mRNA that's read by ribosomes that stay free ribosomes, so they make capsid proteins in the cytosol, and those capsids spontaneously assemble around the genome. And that's another thing, is some of the RNA is copied to make lots of copies of the RNA genome that can be enclosed in these capsid proteins. Then the capsid protein RNA entity buds off with these glycoproteins from the virus, as well as membrane proteins as well. And then it can go infect a new cell. And it's not just one of these, obviously, that comes out, but many, many viruses that come from one infection. So viruses that use RNA as genetic material sometimes are retroviruses. This is not a retrovirus because RNA stays as RNA the whole time. Retroviruses actually take their RNA genome, and they have an enzyme encoded in their genome called reverse transcriptase. So transcribe means to usually read DNA and make RNA. The reverse transcriptase going to take the RNA genome and read it to make DNA. So it's like backwards of what usually happens in the RNA DNA flow. Retro means, backwards kind of. So these are backwards viruses. So the most famous one of these is HIV. After a certain period of time with infection, that can cause AIDS, acquired immunodeficiency syndrome, and that's when your immune system is really not working very well, and people tend to die from pretty simple infections that they just can't fight off anymore. So here would be an example of the retrovirus, specifically HIV. So in here, reverse transcriptase is encoded by the viral genome. That's the little green enzymes. Does the same sort of thing we just saw in the other picture, but this time it has those, it's going to use the reverse transcriptase to read the RNA and actually build DNA. Then it's an RNA DNA double-stranded hybrid, and then the DNA gets a copy of itself, and now you have a double-stranded DNA from the virus. So this is all viral sequence, but that's now made of human, nucleotides, DNA nucleotides, and actually that goes into the nucleus. And this is where a large problem comes, is that it ends up integrating into the nucleus. It's into the genome of the animal cell. It's called a provirus at that point. Now remember, this is a prophage when the phage genome integrated into the bacterial genome. Now the difference here is that the prophage comes back out again when the cell's going to go into the lytic cycle and destroy the cell. In this case, the provirus stays in the chromosomal DNA forever, unless the cell dies. And so there's not going to be a lytic cycle to this viral infection. So what happens is that the provirus, the DNA sequence now of the virus is going to be read when the chromosomes are, expressing genes. Those are going to make mRNA, like the normal pathway in an animal cell. Some of that mRNA is read in order to make capsids, glycoproteins. Some of it's copied, or it's going to be incorporated to make the genome of the RNA, and then the viruses bud off and go infect more cells. And you can see that here. There's the budding part after it's basically made a bunch of copies of itself. So that's a major problem, because they don't go out of the genome after they get in there.
Understanding the structure and life cycle of HIV, the AIDS virus, is critically important to dealing with the disease. HIV is covered by an envelope derived from a host cell membrane. Glycoproteins studying the envelope recognize and bind to receptor molecules on the host cell. A protein coat surrounds the viral genetic material, which consists of two molecules of single-stranded RNA. The enzyme reverse transcriptase enables the virus to make DNA from an RNA template, a trick for which this group of viruses is named retroviruses. The RNA molecules of HIV enter a host cell when the virus fuses with the plasma membrane and the coat proteins are removed by enzymes. Reverse transcriptase catalyzes the synthesis of a DNA strand complementary to the viral RNA strand, and then a second DNA strand complementary to the first. The double-stranded DNA is incorporated as a provirus into the host cell's chromosomal DNA, where it may lie dormant for years. Occasionally, the provirus is transcribed into RNA. This RNA serves as both messenger RNA for the formation of HIV proteins and as genetic material for the next generation of viruses. Protein coats form around viral RNA and reverse transcriptase molecules. Viruses bud from the host cell, acquiring envelopes as they leave.
Okay, so that's a nice little, I think it helps to see it in action. It's the retroviruses that do that, and that's the provirus problem. So the question is, how do viruses make you sick? So, if you're a bacterium, obviously the viruses are going to possibly, if it's the lytic cycle, they end up actually destroying the cell. Same kind of thing-ish can happen in animal viruses. Mostly it causes the lysosomes to release their contents, and some of that can damage the inside of the cell, digest that. And that would obviously cause problems. You also have viruses that produce like let's say it's an integrated part in the genome, or even just whatever's being produced from the mRNA. They can produce toxins. So that's a real toxin, not like toxins are actually scientifically defined as molecules produced biologically that are detrimental to the receiver of the toxin. Anyway, so these lead to disease symptoms because they can interact with processes and make the person very sick. And then the toxic molecular components, like the envelope itself, can be toxic. So just having the virus present at all is going to make the host sick. Viruses are it's kind of funny because they're not considered living, but they do have like those characteristics of life. And so the idea right now, although it's not certain yet, the hypothesis is that viruses evolved after cells, since they can only replicate within cells, and they infect all the different types of organisms that exist, archaea included, fungi, protists, bacteria, and animals and plants. And so what they think might have happened is there would be like some naked nucleic acid, possibly little plasmids or transposon elements that move around in the genome, and those could have gotten incorporated into some of the cells. And if the sequence was a specific way, eventually some of those would have been able to copy themselves, or hijack the host cell in order to get copied, and eventually they just continue infecting. Now, most of them probably didn't do that, the incorporated nucleic acids, but if that's happening millions and millions and millions of time over an extended period of time, you'd expect that at least once or twice that would occur. We actually think it probably happened a lot more times than that. Viruses do evolve. In fact, they'd evolve very quickly, particularly the RNA retroviruses. Retroviruses don't have proofreading when they replicate their genetic material. So when you're building the DNA strands from the retrovirus, there is no checking of the sequence, which means that you get mutations at a higher rate. And so each new round of viruses is a little bit different than the previous round as far as sequence goes. And sometimes that gives the viruses sequences that allow them to avert being killed by drugs. So it's not that the viruses, experience the drug and then come up with a way to circumvent being killed by it. It's that the population of viruses is so diverse already because of this high mutation rate and because there's so many produced in such a short period of time that just necessarily, if you look at 1 million viruses, there's going to be a lot of variation. And so that variation means that some of those viruses already have the ability to, have some way to circumvent the drug. A lot of times it's like, the drug can't bind to the virus anymore because it's glycoproteins have changed, or the enzyme that it might disrupt maybe is a little bit different, something like that. Anyway, you can see resistance is very fast. So here's an example of a study they did with this 3TC drug in HIV patients, and you can see one patient is pretty much resistant like under 3 weeks, about at 3 weeks, and then patient number 2 fully resistant at 8 weeks. So it's a really fast resistance to a drug, which is why they tend to treat viral infections, first of all, not very successfully, and second of all, why they do it with cocktails. So you treat with a bunch of different drugs all combined, and hopefully each of those drugs can target or can destroy different viruses, different versions of the virus. And so if you have a bunch of different, assault strategies on the virus, maybe you kill all the population instead of having survivors because of their differences in genetics. So here's just an explanation of what's happening, and you can read through that on the notes. We can also track viral sequences now, and this is an example of the looking at the H1N1, the influenza A in Taiwan, and so you can look at this in your book. You also have an exercise on this in Mastering Biology, but you can just see all these branchings show which viral strains derive from the same original virus, and then they become so different from each other they can be even considered different groups, and you see those groups are all clumped together because they come from the same viral parent, which the next generation had, 1, 2, 3 different mutations. And then this viral parent, if you want to call it that, came up, some of its offspring would have 2 other different types, and so it would branch off and branch off. So how do you prevent viruses or get rid of them? Mostly prevention is the way to go, because our antiviral drugs on here are not very effective. Sometimes they can treat symptoms, but they're not very good at getting rid of the viral infection. You can't treat with antibiotics. Antibiotics are specifically against bacteria. So if you have a cold or most respiratory infections, taking an antibiotic like a Z-Pak or something is not going to work. Usually it seems like it works probably because you're at such a far point in your viral infection that you get better pretty soon after you start taking it. It's actually a really bad idea to take antibiotics when you have a viral infection because the bacteria present in your body normally, of course, get acted upon by that antibiotic. You can kill good bacteria, or you can also select for bacteria that are resistant to that antibiotic, meaning that they have a DNA sequence just naturally that they don't get killed by the antibiotic. And so maybe one day when you need to kill those bacteria, the antibiotic doesn't work anymore, because most of the bacteria in your body are ones that are resistant because you took that drug, especially if it's repeatedly, when you didn't need it. So what we can do, though, the prevention part is vaccines. Really, really important to vaccinate yourself and your children and get your boosters. That's how we basically cut down on the amount of deaths from all the different viral infections. And so what happens is you can't actually get a viral infection from a vaccine. They are what we call attenuated viruses, which means that parts of their genome have been removed so they can't replicate, or you just put parts of the viral proteins into the, inject them in the body, so it's not even a whole virus. And what they do is basically expose your immune system to seeing the viral parts. And the immune system, the way it works is that it would produce antibodies and have like a memory of that infection and which antibodies would match it. And then if you get the infection that can trigger this production of antibodies it's already seen very quickly. And so the body can fight the infection off before it takes hold and you don't get the viral infection. So viruses, there was that whole rumor about autism and viruses, which if we just look at thousands of studies, we know that that's just not the case. You can't overload with viruses. You do have to, if you get quote unquote sick from a virus, there's one of 3 things happening. 1, you've already gotten the infection and you've gotten a shot too late, like let's say the flu. 2, you have some soreness or redness or inflammation that's from actually the shot itself, so like, poking and breaking the skin. Or 3, you're allergic to some component of the either the way the viruses were made. A lot of times they're made and they end up having albumin in them, egg protein. So if you're allergic to eggs, you really need to tell them. They have other ways of the viruses, like through your nose. And so that would be the other option of why you might get sick, but it would not be from the virus itself. That can't happen. And so these viruses that we have to worry about today and are making vaccines for very quickly are emerging viruses. So they're ones that like pop up out of nowhere, like HIV or West Nile. We know about that here. It actually wasn't even seen in the U.S. until 1999 in New York, and now it's except for Hawaii and Alaska, it's in every single state in the country. And we call a general outbreak in a localized area. That's what an epidemic is. So an epidemic, for example, would be H1N1. That was localized, at least initially, to Mexico and the U.S. So that was an influenza A virus back in 2009. You might remember that one. It was the one That's the swine flu one. The reason they call it that is because it might have jumped from swine over to humans, not from what the swine used to, the virus that infected the swine, but some kind of combined virus that had the ability now to infect humans. But it's not like, yeah, okay. Anyway, so H and N, when they name those names, the H part is for one type of protein, hemagglutinin type 1, hence the 1. That's going to help the virus attach to the cells. And then the N part is the neuraminidase type 1, since the N1, and that's going to help virus, or the enzyme that helps to release the new viral particles. And this is a picture of what that H1N1 virus looks like. So how would you get this emerging viruses to happen? There's 3 different ways you need to know. So the first way is, I mentioned the mutation rate's very high, and that's when you get new viral strains. RNA viruses don't have proofreading. With these new mutations, the population hasn't seen that combination before, so they have little resistance, and you get infections spreading very quickly. In the another way would be when you have isolated populations and now people are exposed to those groups, whether they start traveling, because now we have, so much easier travel to all kinds of places in the world. And, or like if you build roads, through somewhere that was isolated previously, now you're getting exposed to whatever that human population has. And that can spread into the groups of people that have never seen it before and spread very quickly because there's no immunity to that. And then number 3 would be that the viruses come from other animals. A lot of times the animals aren't even affected by it, so they wouldn't show symptoms. It's basically what happens is they get infected probably by a number of different strains. Viruses can recombine their DNA, so they mix it up between different viruses, and then what comes out of that viral reproduction, if you want to call it that, is going to be a different strain, and sometimes that has the ability to now infect humans because of the different proteins that are encoded for, and that would be how it would jump from, let's say, a pig, swine flu, or the avian flu, bird flu, H5N1, how it would jump from one organism into the humans. There are some viruses that actually infect more than one organism. They call them zoonotic diseases or viruses. So those would actually be ones that an animal would carry that could pass directly to a human and affect both of them. So we said epidemic is a general outbreak. Pandemic is a global epidemic. So now it's all over the place. So the Spanish flu is a really famous one. 40 million people died from that in 1918, 1919. Pretty intense time period, and they really didn't have anything to do for that. I guess they'd drain your blood sometimes and basically just wait it out as best as they could. And then, 1997, we have the avian flu, the H1N5, more properly. It had a really high mortality rate. I mean, it's still around, but I think it was killing more than 50% of the people who were getting infected by it, which is really high. But it's not as easily spreading. We do have to worry about that, mutating to gain qualities that allow it to spread from human to human. And then the H1N1 in 2009, also killing young people. Usually old people are the ones that are affected most and most often, and also spreading very quickly from place to place. So here would be what they look like. So this is the H5N1, this is the H1N1, those are the little blue dots. And then, they had different types of screening, and probably back in 1918, not the best strategy for containing viral infections, having everyone in one huge room, but, there was little else to do. You can see some people with masks on, which if you have the right type of mask that has the small pores, that can help prevent viral infection. I'm going to post this actually, Discovery Channel. This would be important to watch about the emerging viruses. I'll probably, let's see, it's only like, I think it's only like 3 minutes. So why don't you take a look and go watch that. I think it's, I'll ask you a question about that idea on the exam. And then finally, just to point out that plant viruses are extremely prominent. Lots and lots of types of viruses, many more plant viruses than animal viruses. So when you see plants with lots of spots on their fruit or their leaves, a lot of times that's a viral infection. Can't do very much about it, unfortunately. And it cost us $15 billion annually worldwide in food crop losses. So it's a big deal. And so this would be an example of a squash with a viral infection. You can see the black is not a typical, how typically the squash looks. And it's kind of interesting. So just like, we think of viruses horizontal transmission, so viruses in plants can get passed from one another through, cell wall entry. And there's also vertical transmission, so actually that the virus hangs out in the parent and actually gets passed on to the offspring. We don't have that, as far as I know, in humans, but this is a big problem in plants. I also want to point out, actually, that you know how you get some viruses coming back, like especially, let's say, herpes virus. So, you get the cold sores or the genital sores that flare, right? Chicken pox is when you're young, and then it comes back as shingles when you're older. If you have mono, you might get it. It might, flare up again. And the reason for those is that not like a provirus where it's in the DNA itself, but those kind of herpes viruses actually will have little pieces of viral DNA that hang out in the nucleus forever, and they get replicated with when the cells get replicated. And so every once in a while, if there's an environmental stressor, those pieces will get expressed, and then you'll have the viral infection flaring, like it'll start producing toxins. The DNA will be red and the toxins will be produced, or possibly the cells, the lysosomes start killing the inside of the cells or something like that. So that would be why you flare. It's because those guys are hanging out there kind of dormant a lot of times for an extended period of time until there's an environmental trigger for them to turn on their gene expression. And so that would be, response to the environment. I talked about this in the other video, so I'm not going to do it all again. But the response to the environment is definitely a characteristic of viruses, that they, in the lysogenic lytic cycle, when they can go between the two, that's its response to the environment, as well as these kind of flares that you get with the chickenpox shingles or the cold sores and that kind of thing. And then I wanted to point out that I put up this chart online where hopefully you can look at all the characteristics of life on the left, and then write a little thing to help organize your ideas about living organisms and how they show that characteristic, because they all should. And then viruses, and if they share the characteristic or show the characteristic or not. Because some would, like reproduction, yes, but can't happen on its own, which is like the big deal, right? Energy transfer and transformation, they're not doing any type of metabolism, meaning not taking any energy in. They don't eat. They don't produce any ATP themselves. So that's not going to be a characteristic. We just said response to the environment, that would be a yes. And you could give an example of the transition from lytic cycle or lysogenic to lytic cycle. We definitely said evolutionary adaptation occurs in viruses, and I showed you that. Clearly not made of cells, and etc. You could go, on there. They are ordered though, right? So it's not random accumulation of materials. So that's what you're going to want to look at for the rest of chapter 17. Let me know if you have any questions, and the Mastering Biology should help you as well.